Good afternoon, everyone, and greetings from Davao City, Philippines. This afternoon, I'll be presenting our paper entitled, let me share screen, Deep Structured Populations of Geographically Isolated Nipa fruticans worm in the Indo-West Pacific revealed using microsatellite markers. First, what is nipa? Nipa or Nipa fruticans worm belongs to the family Ericaceae and a monotypic subfamily Nipoideae owing to its distinctive morphology and habitat specificity. Nipa as an underground horizontal stem that has the ability for dichotomous branching. And Nipa populations occur in areas that are regularly flooded with brackish water brought about by tidal action. Nipa is not only considered important ecologically, but economically as well because it provides livelihood, such as source of thatch, and sap from the peduncle or fruit stalk. The sap is used to produce fermented beverage called tuba and vinegar. The sap is also processed into Nipa vodka with 70 to 100 proof. That's 35 to 50% ethanol using indigenous distillation process. So Nipa will most likely be a good source of bioethanol in the future. In terms of population genetics, different conclusions have been drawn in different geographic regions using different markers. In fact, some studies would suggest that Nipa populations did have genetic variation. By including larger areas from 6 populations in China, Vietnam, and Thailand, polymorphisms were detected in 2 out of 17 SSRs. In fact, Lapitan and Nicolas reported considerable genetic diversity from the accessions they collected from southern Luzon in the Philippines using the 25 polymorphic SSRs. So we pursue the use of microsatellites or simple sequence repeats or SSRs as molecular markers because they are not only popular, but are also useful in population studies and evolutionary processes. So these are our sampling sites in the region. We have Philippines, mainland Southeast Asia, consisting of Indochina and Malay Peninsula, Indonesia, and lastly, Sri Lanka. So these are the 18 polymorphic SSR loci that we use to evaluate the 445 Nipa samples that we collected from the Indo-West Pacific. In terms of genetic diversity, the observed heterozygosity, as indicated by HO, was lower than expected heterozygosity, and estimates of F as measure or coefficient of inbreeding were high, particularly in Sri Lanka, where the levels were extremely high, likely due to inbreeding. So by looking at this data, we already have the clue that populations in the region were genetically structured. The analysis of molecular variance indicates that over half of the variations was coming from among individuals within population. Statistically, all levels were significant. The effect of isolation by distance, or IBD, was detected statistically significant in Mantel test with a positive correlation R equal to 0.4199. So this would indicate that individuals that are close to each other tend to be genetically similar than those that are far from each other. To analyze the relationship between genetic divergence and geographic distance, we conducted Mantel correlogram as an extension of Mantel test. So W here is the distance class, and this red line divides the negative from positive correlation. So the field black square would indicate statistically significance, and that the unfilled square is not significant. So the shape of Mantel correlogram generally shows a trend that was decreasing significantly. So at distance class 8, that's about 8 or 2,000 kilometers apart, a pair of samples was differentiated with significant negative correlation and no spatial correlation between distance class 13 and 14. So taken together, the spatial analysis suggests a significant genetic divergence of individuals and structured populations with geographical patches of nipa in the region. So when samples are plotted across space by the principal coordinates analysis using the kernel density, that is to assess the distribution of genotypes onto the principal axis, one larger group belongs to Southeast Asia and a smaller group to Sri Lanka. This pattern was also observed using the multivariate discriminant analysis of principal components, which also showed two genetic clusters, Southeast Asia grouped in one cluster, while Sri Lanka in distinct separate cluster. However, we observed some samples grouped with Sri Lanka coming from the northern coast and southwest, which were likely effective migrants or hybrids between these two regions. So one novel approach in determining spatial structure is called the spatial principal spatial principal components analysis, and the ones that we marked on this map basically corresponds to the shape of the polygon that consists our sampling sites. So overall, there's a strong connectivity network was observed around South China Sea, and this being indicated by this intense red background, except for southern coast and southwest, along with the Indonesian population, where they were less associated with the South China Sea populations, while those of Sri Lanka were distinctly separated as indicated with this blue background. So Monmonier's maximum difference algorithm was implemented to evaluate a genetic barrier, and the results showed that Malay Peninsula is a genetic barrier as indicated by this blue solid line. So a more detailed analysis of the genetic structure in the region was conducted by using the package GeneLand, which runs a multi-locus data set with coordinates. So if the inferred number of cluster is equal to 4, so this is the result of the GeneLand, which is being interpreted on this map. So populations from the Philippines belong to one group, while most of the populations from mainland Southeast Asia belong to another group, except for southern coast and the southwest population, because they are grouped along with Indonesia, while Sri Lanka was distinctly separate from the rest of Southeast Asia. This is in terms of spatial model. That means we consider population membership. In non-spatial model, that means we did not consider population membership. The inferred number of clusters is equal to 6, and except for Sri Lanka, the populations or some populations would belong to group of other population in the sub-region in Southeast Asia. So using the structure again, Sri Lanka was distinctly separate from Southeast Asia when K is equal to 2, and if K is equal to 3, the additional red cluster are found across Southeast Asia. And if K is equal to 7, the Southeast Asian populations are clustered into 6 groups, particularly the Malay Peninsula, which is pretty admixed, and we consider it as melting pot. So in terms of the phylogeography of nipa, the ancestry coefficient analysis showed the genealogical lineages of nipa populations can be categorized into Sri Lanka and Southeast Asia for k is equal to 2. For k is equal to 3, Southeast Asia can further be subdivided into mainland Southeast Asia and Philippine archipelago, where Indonesia shared with. The island of Sumatra was projected to share ancestry with Sri Lanka. When k is equal to 7, the genetic divergence of Luzon West and Vietnam 1 shared a putative ancestry with the southeastern coast of Malay Peninsula. So the sharing of these lineages is a clear indication of migration as influenced by oceanic circulation. The founder effects is expected if the individuals migrated from mother populations such as Southeast Asia to a new niche further away. So if Sri Lanka was genetically differentiated from Southeast Asia by isolation by distance, then it must show by the tests using the software bottleneck. So based on the graphical descriptor, Sri Lanka were all mode shifted, meaning distance experienced bottleneck recently or sometime in the past. To graphically evaluate the genetic clusters of nipa population, a phylogenetic tree was reconstructed by neighbor joining method. Again, the result indicated that the genetic distinctiveness of populations from Sri Lanka, which was further clustered with Malay Peninsula. This further confirms the genetic connectivity between Southeast Asia and Sri Lanka in certain populations as observed in the APC. So this tree is represented by the unrooted phylogram, meaning that the root is not hypothesized. Nevertheless, the deeper nodes of the phylogenetic tree have low bootstrap support. Notwithstanding the lines of evidence of the founder events in Sri Lanka by oceanic circulations, there are indications of stronger genetic link eastward rather than westward. For example, this purple circle is representing Matara, had a strong genetic link with Southeast Asian populations based on the contemporary genetic diversity. This pattern is considered recent migrations where inbreeding with local populations in Southeast Asia diminished their ancestral genetic lineage that resulted in low bootstrap support. So this is the proposed recent migration routes of nipa, and hypothesized that their potential entry point from Sri Lanka to South China Sea might be the Sunda Strait. Okay, so the dashed red arrow is the migration pattern, indicate migration pattern from Sri Lanka, and this red dashed line box is the hypothetical route to northwest Sumatran population by long distance dispersal to Southeast Asia via Sunda Strait. These blue arrows are the prevailing surface currents during southwest monsoon, and these red solid arrows indicate strong migration links during our simulation. So those are the results of our study, and thank you for listening again. Good afternoon.